Dear students, welcome to my online classes. In previous class we have discussed about the introduction to insulin. Today we will be discussing about production of human insulin, that is production of humulin. Humulin is nothing but human insulin. In the previous class I told you the insulin given to human beings from other animals, extracted from other animals, used to cause many allergic reactions and adverse effects. In order to overcome that, in the year 1977, Itakura chemically synthesized the human insulin, but that also was a very tedious process and very time-taking process, tedious process, and as well as the costly process, and also it was not so accurate as human insulin. So later, the scientists thought of producing human insulin by using genetic engineering technique, by using biotechnological techniques. So how we do it? Let's see. Right? Clear? Production of humulin or human insulin. So we all know that in the previous class I have told you discuss about the beta cells, pancreas, and all those things. So we have one gene, one protein concept. Insulin is basically a hormone, but basically a protein. You all know protein synthesis takes place. In a previous chapter of biotechnology, you have studied about plasmid. What is plasmid, what is recombinant DNA technology, and how it is being done by using restriction enzymes, and what is cloning, what is recombinant DNA, what is a plasmid, fine, what is a cloning vector, what is a bioreactor, all these things you have studied on that. So the application part of that we have to study here. So here the very first step is isolation of proinsulin gene. Now why proinsulin gene? Because whenever it is produced in our body, it is produced as proinsulin. All those the background and the introduction part is over in the previous class. So we have to isolate the proinsulin gene. Now where this insulin is produced? In the pancreas. Who produces? Beta cells, islets of Langerhans of pancreas. So where we get this gene? From beta cells. So isolation of proinsulin gene is our heading. So here from beta cells of islets of Langerhans from pancreas we have to isolate the gene. So in order to isolate the gene, what we do? We isolate mRNA. What we isolate? We isolate mRNA. Now when we isolate this mRNA is treated with reverse transcriptase enzyme. What enzyme? Reverse transcriptase enzyme. When you treat this mRNA with reverse transcriptase enzyme, this mRNA undergoes a reverse transcription process that you have studied in molecular biology chapter. When mRNA undergoes reverse transcription process, it produces DNA. What it produces? DNA. And that DNA we call it as complementary DNA. What DNA? Complementary DNA. Since DNA is being synthesized from RNA, we call this process as reverse transcription. That's why the enzyme what we use here is known as reverse transcriptase. All the RNA viruses have this enzyme. Like HIV also have this enzyme. We have discussed in human health and diseases when we are talking about AIDS and HIV. So reverse transcriptase enzyme, it brings about reverse transcription. This process is also known as timinism. Timin is the molecular biologist who find out this process. Hence reverse transcription is also known as timinism. Timin in Baltimore, I have told you earlier also. They were the molecular biologists. So from RNA we have produced DNA. This complementary DNA is known as complementary DNA. Now this complementary DNA is nothing but what? Proinsulin gene. What is known as? Proinsulin gene. This complementary DNA itself acts as we consider this complementary DNA as proinsulin gene. I hope it's clear. After that we have to insert this. Second step, insertion of proinsulin gene into plasmid. You all very well know what is plasmid. Extra chromosomal, double-stranded, circular, self-replicating bacterial DNA. What is known as plasmid? and now this plasmid we are taking from E. coli. What coli? Escherichia coli bacteria. E. coli bacteria. Without E. coli, microbiology and molecular biology is almost impossible, because we find a lot of application of E. coli in medical microbiology, in molecular biology as well. So what we are doing, we have isolated in the first step. Now we are inserting it. How to insert? You all know it's a plasmid. Plasmid into plasmid. Plasmid of what? E. coli bacteria. Now this plasmid of E. coli bacteria should be treated with REN. What is REN? Restriction endonuclease. Restriction endonuclease. And now it is used. This plasmid is employed by restriction endonucleases. They cut the DNA segment. Similarly, same REN is also used to cut this complementary DNA. Why the same REN? We are using it here because you have studied in the 11th chapter. They are specific in their action. All those things, mechanism of REN also you might have studied. So here, to get sticky end, same REN is used here. Same REN is also used here. Restriction endonuclease is used here. In order to get what? Sticky ends. Why sticky ends? So that we can combine them. We can join them by using what? Yes, which enzyme? For binding, joining, ligase. What enzyme? Ligase. So here, by insertion of proinsulin gene into plasmid, this is a plasmid taken from E. coli bacteria. E. coli bacteria. This plasmid is also treated with restriction endonuclease that cuts at the specific site, and then this complementary DNA, what we consider it as the proinsulin gene, is also treated with same restriction endonuclease, so that it makes complementary edges or pairing complementary sites, and also what we call as sticky ends, so that we can join them. After that, this is taken, this cut plasmid is taken, and this DNA is taken. Then what is this? Making the recombinant DNA. Why we call as recombinant DNA? Because we are combining this. What is this plasmid? Is nothing but a DNA, and this DNA is taken. So here it is like this. Fine, it is like this. Fine, then. So here this DNA will be there. This has to be binded here by using ligase enzyme. Ligase is used for ligation, the process of binding, joining, combining the different genes. When one gene is added into a DNA, that DNA is called as recombinant DNA, and the technology is known as RDT, recombinant DNA technology, or we can call it as genetic engineering also. So how do we do it? You have studied in the previous chapter by using cold calcium chloride and heat treatment. What treatment? Heat treatment. So this plasmid and the complementary DNA gene are taken into cold calcium chloride solution. This cold calcium chloride solution, fine, softens the plasma membrane of the E. coli. Sorry, just after you forget this, the next one is insertion. What is this? Insertion of recombinant DNA into host. Host is which one here? Again, don't get confused. Host bacteria is also again E. coli. Here by using ligase enzyme, you have inserted this proinsulin gene into plasmid. Now this plasmid DNA, what we call as recombinant DNA. Now why is recombinant DNA? Because it is recombined. We have added one more gene, the desired gene, the needed gene, the desired gene. Which is the desired gene here? Proinsulin gene. Once this recombinant DNA is ready, we have to insert this recombinant DNA into what? Into the host cell. You have studied as competent host in 11th chapter. That competent host is again here E. coli, because the plasmid is also taken from E. coli. How do we do it? This way. What is the method? By treating it with cold calcium chloride. This cold calcium chloride softens the plasma membrane of the bacteria, of the host. When once it is softened, immediately after 1 or 2 minutes, in that chapter you would have studied, 1 or 2 minutes it is treated with, 3 to 4 minutes also you can treated this with cold calcium chloride that softens the plasma membrane. Immediately after cold treatment, treatment. It is given heat shock, what we call it as heat shock method, where it is treated with 42 degrees Celsius or slightly more than 42 degrees Celsius. Why? Because it is a mesophilic bacteria. E. coli is a host. Here also we are using E. coli is also a host. E. coli is a mesophilic bacteria. What do you mean by that? The bacteria that can survive between 20 to 40 degrees Celsius. That is what is known as mesophilic bacteria. Above 40 degree, if you heat them, they rupture. So here, when it is heated to 42 degrees Celsius for about 1 or 2 minutes, for about again 1 or 2 minutes, fine, 1 to 2 minutes, the rupturing of the plasma membrane takes place. Once it ruptures, this has this plasmid DNA, or what we are prepared now, the recombinant DNA, has affinity towards the cell. Which cell? E. coli. What is this host? So it gets into that, it enters into that. So this is the E. coli cell. E. coli cell. We have made recombinant DNA. We are heating it. We are cold. First of all, the first step, cold softens the plasma membrane. Softens. After that, immediately heat shock is given. Heated. Plasma membrane ruptures. When through that opening, through that holes, through the rupturing, this recombinant DNA enters into the E. coli. Now this E. coli is a recombinant cell. What is it? Recombinant cell. What we are using as host. Recombinant means competent host. The host, it has which gene? Human insulin, proinsulin gene. Taken from whom? Human beings. Don't get confused. Our own gene is taken out. Since our own cells, beta cells, cannot produce insulin, but genes are there. Genes are normal. They are not expressing their function. So our own gene is taken, inserted into bacteria. Bacteria produces that insulin. Very simple concept. There ends the matter. Now after that, competent host. You have studied bioreactor. Now this host is taken. Production. Now what is it? Production of the insulin or proinsulin, you can say. Production process. How do we produce? By using bioreactor. What is bioreactor? You might have studied the definition. 1000 ml stainless steel furnace, like a cylinder-like structure where media is taken, temperature is maintained, pH is maintained. These host cells are taken and they are subjected to 6 to 8 hours of process. During this process, what happens? Since we are providing all the optimum conditions, all the required conditions like temperature, pH and nutrients, this bacteria which has this host, this bacteria E. coli, which has our desired gene, our insulin gene, multiplies. When this bacteria multiplies in bioreactor in a matter of about 4 to 6 hours, when it is multiplying, what's happening in that bacteria? The gene what we have inserted is also, yes, what happening? Multiplying. That's also multiplying. What do you mean by that? So when it multiplies, it expresses its function. It produces what? Proinsulin. Proinsulin is produced in the bioreactor. In bioreactor, you would have also studied downstreaming process. After the product is produced, it has to be purified. For purification process, we go for downstreaming process. Purification. What is downstreaming process? The process of separation and purification of the product produced in the bioreactor is what is known as downstreaming process. The definition you would have studied in the 11th chapter. Fine. So purification process. Now what is purification? We have to purify this process. Why? Because it is produced. Which it has produced? It has produced proinsulin. What is our target? To produce insulin. You know the difference between insulin and proinsulin. In the previous class we have told. Proinsulin has 86 amino acids, 86 amino acids. This one has insulin has 51 amino acids. The C chain has to be removed. Now this has A chain. What is the purification process? A chain 21 amino acids, B chain 30 amino acids, C chain 35 amino acids. Altogether 86 amino acids. It is a impure form, non-functional. It cannot perform its function. Clear? And one more point I have to add here. When you are adding this by using ligase, you know one factor. What is that? Selectable marker. So here selectable marker is fine. Beta galactosidase near LAC-Z gene. I will write here only. You can repeatedly watch this class. LAC-Z gene you have studied in molecular biology also. It produces beta galactosidase enzyme. It can be asked in NEET exam also. Beta galactosidase is also used as a selectable marker. Right? So near the selectable marker is tagged. Now this is this is recombinant gene, proinsulin gene. It is tagged beside, adjacent, beside the laxate gene. We are ligating here by using ligase enzyme. Why? In order to see the expression of this gene. When beta-galactosidase is produced in the bioreactor, that indicates that says that proinsulin is also being produced. Why? Because this product is also there. Which product? Beta-galactosidase. So along with the proinsulin, what is that? Beta-galactosidase. Our aim is not to produce beta-galactosidase. Our aim is to produce insulin. But correct, insulin is produced in the form of proinsulin. We have to purify it. So how first we have to remove this beta-galactosidase? How it is being removed? Very important point. By using cyanogen bromide. What is it? Cyanogen bromide. When you add this after the production of proinsulin into the bioreactor during purification process, you add cyanogen bromide. What is the cyanogen bromide? It has the affinity towards beta-galactosidase. It goes and combines to beta-galactosidase. Now what is here? Proinsulin and beta-galactosidase is here. Like one product, you are adding cyanogen bromide to this. Cyanogen bromide goes and binds to beta-galactosidase. So it separates. Only proinsulin comes out. After that also, again this proinsulin, our aim is not to produce proinsulin, our aim is to produce insulin. So how to remove this, how to make this proinsulin to insulin? By using an enzyme known as carboxypeptidase. What is an enzyme? Carboxypeptidase. What is carboxypeptidase? This enzyme removes the C chain, It removes C chain. Thereby, from 86 amino acids, it will become to 51 amino acids. That is nothing but our insulin. So, by using carboxypeptidase enzyme, artificially carboxypeptidase is used in our body. Question may come to intelligent students: how it happens in our body? Because in our body also, proinsulin is synthesized, proinsulin is produced whenever it is required. Functionally, whenever we require, the proinsulin gets converted to active insulin by using trypsin enzyme. Remember, trypsin is also secreted by pancreas, other part of pancreas. That is nothing but the exocrine part of pancreas. Trypsin and chemotrypsin you might have studied. So here, either trypsin can be used or carboxypeptidase can be used. Now once you remove C chain, removed. Now this A and B chain has to be combined by using disulfide bonds. By using disulfide bonds, A chain is attached with the B chain. So this A chain, B chain are attached. Now this becomes a functional insulin, exact insulin. Human insulin is produced by using E. coli bacteria, by using biotechnological processes, Indian genetic engineering processes. Hence it is known as human insulin. This was first produced by Gene-Tech Biotechnologies in 1978. Later, the procedure was taken from another biotechnology company from USA, that is Eli Lilly. In 1982, they produced it, but they named it as Humulin and marketed in 1983. Eli Lilly is the first company to market. Gene-Tech is the first company to produce it. In our country, in India, Biocon is the first country to produce human insulin in the form of humulin. Human insulin only produced in bacteria. Again, after purification process, given back to the human being. So human beings don't suffer from any side effects because it is human insulin, very similar to natural insulin. I hope it's clear. Repeatedly watch this class, very important class. Study refer the study material and study textbooks also, NCERT books and other books also. Keep studying. God bless you. Good luck. Take care.